Hello, everyone. My name is Hanif Ahmad Kausar Junaidi from the Research Center for Care and Control of Infectious Diseases, Universitas Pajajaran. And today, I want to share with you the findings of our study entitled The Global Biogeography of Mycobacterium Tuberculosis Indo Oceanic Strains from Whole Genome Sequencing Data. So Mycobacterium tuberculosis, the Indo-Oceanic strains, or more colloquially known as ancient lineage 1 or just lineage 1, is prevalent in many countries around the Indian Ocean, and this strain is the most dominant strain in Indonesia's eastern provinces like Papua. However, our understanding of the spread is limited, and thus, in this study, we attempt to describe the geographic spread of MTB lineage 1 using whole genome sequencing data. So a total of 306 whole genome sequencing data of MTB lineage 1 from all around the world were identified and downloaded from the Sequence Read Archive and the European Nucleotide Archive. The data underwent bioinformatics analysis using TB Profiler, from which we obtained sublineage and drug resistance mutations information. And then in parallel, these data also underwent bioinformatics analysis using the Phoenix pipeline, the results of which we used to reconstruct a phylogenetic tree using the maximum likelihood method. The tool used here is called IQ tree. So here are the global distribution of the data set that we downloaded. So a majority of the data, about 48%, came from Southeast Asia. Next is from South Asia with 17%, and then various other data sets from other regions of the world, North Asia 5%, West Asia 5%, Africa 7%, Europe 5%, North America 7%, Oceania at 3%, and South America at 2%. From the results of TB Profiler, we obtained the drug resistance associated mutations. So a majority of the isolates that we obtained, 72%, are sensitive to anti-tuberculous drugs. About 28% have at least one mutation that is associated with drug resistance, and a total of 9% had mutations that are associated with multi-drug resistant TB. From these drug resistances, a majority have mutations that are associated with isoniazid resistance, about 19%, followed by ethionamide at 15%, rifampicin at 10%, followed by ethambutol, streptomycin, and pyrazinamide, and then a total of 9% had various other mutations that are associated with other drugs, that are associated with resistance to other drugs. This is the results of the phylogenetic tree reconstruction. The annotations of the inner color strip are the region of origin, and then the outer color strips are the sublineage information. So just based on appearance, we can see that there are three large monophyletic clades based on the different sublineages. So at the top here, we have a monophyletic clade based around lineage 1.1. At the bottom left, another large monophyletic clade is based around the lineage 1.2.1, and at the bottom right is a monophyletic clade based around lineage 1.2.2. And also based on appearance, there appear to be a regional divide based on these clades and sublineages. So for example, there seems to be a lack of isolates from South Asia in the clade 1.2.1, and in clades 1.1 and 1.2.2, there seem to be a lack of isolates from island Southeast Asia. So from here, we mapped the geographic distribution of the sublineages. And from here, we can see that in Africa, West Asia, South Asia, and mainland Southeast Asia, the proportion of isolates from clade lineage 1.1 seem to be the dominant sublineage, while in island-based Southeast Asian nations, North Asia, Oceania, Europe, South America, and North America, the isolates based around clade lineage 1.2.1 is the dominant sublineage. And in Africa, West Asia, and South Asia, there appears to be a high proportion of lineage 1.2.1 isolates. So in conclusion, there was significant difference in sublineage profiles between regions, particularly in the proportion of lineage 1.1 and lineage 1.2.1. However, further study is needed to determine the route of transmission of MTB lineage 1 into Indonesia. So above here, we have the sources that were used to download the global data sets. And at the bottom are the bioinformatics and statistics tools that we used. Thank you for your attention and thank you for listening. And I hope that you find this presentation informative.